From Microbe TV, this is Immune, episode number 94, recorded on June 17th, 2025. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back. How are you?
Well, let's see. It's still raining. It's been raining for 5 days here.
It's been raining for 5 months.
5 months.
Forget 5 days. We did have a nice weekend here, but then it started raining again. Yeah.
Also joining us from Cleveland, Ohio, Steph Langle.
Hi, everyone. Great to be back. And yeah, we have rain. Lots of rain. We had like a river in our front yard the other day.
A river
Popped
runs
out of nowhere.
And from Madison, New Jersey, Brianne Barker.
Hi, great to see everybody. It's raining here too.
Oh,
surprising to no one.
Get your arcs ready.
Yes.
Yeah. Or at least a kayak.
Two of each species. Now, you couldn't ever do that, right? Because there are too many species to fit.
Gosh, there'd be a lot of species. Does that also, is it all the pathogens? We got to make vaccines, so we're going to have to stockpile all the viruses on our ark.
I mean, all the viruses,
that can be really
bacteria.
tricky. How do you have all the viruses infecting all the organisms and have the organisms all live?
That ark would be a mess.
Well,
and what about ticks? What about ticks? What about fleas? They'll be jumping all over the place.
Let's leave the ticks out of it.
Oh.
They can stay off. They can stay off the ship.
You can leave them behind and let them.
I don't think they're
actually, they'd probably survive the floods.
They probably would.
It's just ticks left when we're gone. It's just only ticks.
Ticks and cockroaches. Yes. Yeah, exactly.
Yeah. Ticks and cockroaches. And maybe fungi too, because they're pretty hardy.
That's true.
And those little cute tardigrades.
Oh, yeah.
Tardigrades too. Yeah, they'd probably survive. Well, if you like these discussions here that we have, including the silly things we talk about, so we can relax and, get ready to talk about serious stuff, we'd love your financial support. This is Microbe TV, is the company that makes all these podcasts. Science podcasts, by the way, they're about science. We don't have an agenda. We just want to tell you what the papers say, what the science says. We don't lie. We don't misinform you. So if you like that and you want it to continue, we have like 9 different science podcasts that you can pick from. Go to microbe.tv/contribute and help us out. It doesn't have to be huge. If everyone contributes a little bit, we'll be all set and we can continue to go forward and build our arcs, which could be a metaphor for, holding up the bulwarks against the assault against science. We're building arcs.
Yep.
Today we have 2 papers about cells, cells of the immune system. So we'll start with Brianne's snippet.
All right. So I have a paper today that was published in Nature earlier this year. The title of the paper is Granzyme K Expressing CD8 Positive T Cells Promote Recurrent Airway Inflammatory Diseases. There are 4 co-first authors, Feng Lan, Jizu Li, Wang Xuan Miao, and Fei Sun, and one senior author, Hai Kui. And most of these individuals are from institutions in Beijing, like the Capital Medical University, the Laboratory of Nasal Disease, the Institute for Immunology at Tsinghua University, and a few other labs in Beijing. This paper is actually one of a couple of recent papers that came out looking at Granzyme K. So there were 2 papers that came out in Nature right around the same time. There's also a really interesting sort of news and views in Nature Immunology that talks about both of them together. This one, like I said, is about recurrent airway inflammatory disease. The other one is more about rheumatoid arthritis and joint disease. And here the authors were looking at people who had recurrent nasal polyps and recurrent airway inflammation. And they had some patients who had multiple surgeries where they had nasal polyps removed. And they wanted to look and see if there were shared T cell clones in the samples that were taken, with the idea of assuming that this would mean that there were sort of persistent or memory CD8 T cells or T cells that might be involved in causing the disease. And so if the exact same T cell receptor genes were being used over time, then perhaps that's the same lineage of T cells that are causing this disease.
And would you have thought that that was that they would be recognizing a pathogen or recognizing commensal microbes in your nose? Do you have any thoughts on that as you started to read this?
I guess I was thinking that they would be responding to some kind of maybe commensal microbe, but I'm not sure I was completely certain on that.
Yeah, I don't know. It's an interesting thought.
Yes.
But could you, knowing the T cell receptors, could you know what they're recognizing, or is that not possible?
Yes. And
to some extent, because there's now libraries, right?
Yeah.
There's databases where you can go and search and see whether a TCR sequence matches anything known, whether it's been matched before, and they're building those continuously.
They do kind of do a little bit of that and throw it in one of the supplementary figures, but they just kind of say, there's a bunch of antigens. So they don't. We
can't, I don't think we have the technology yet to say this is a TCR sequence and guess what it recognizes. So
no, I don't
there's
think
been
so
some
either.
that have been matched up. There's a growing database, but we don't have enough information to be able to do that yet.
Sounds like a job for AI.
It could be.
You give it what we know and say, predict the structure of a peptide that would fit in here. It's hard, but I think with enough data, as you say, eventually you could do it.
Yeah, I think that could be true.
I mean, now you can predict three-dimensional protein structures, right?
Oh, yeah.
That's so useful with AlphaFold.
Yeah. Yes. I mean, you could do it for many different pathologies. If there was a particular epitope on a cancer cell that a T cell would recognize, that'd be hugely beneficial.
Oh, yeah.
So they didn't do some of those things here. But they did look at these patients, and they found that there were persistent clones within individuals, even though there was a lot of variation between individuals. So it did suggest that there were persisting clones, and there might be some T cell memory going on here. And they also were able to look at the patients with nasal polyps versus healthy controls to look at functional clusters of the T cells to see what kind of T cells they were looking at. They sort of expected to see Th2 cells or type 2 type of immune response with eosinophils and IL-4 and IL-5 and all of that, because that's what we often think about with allergic inflammation. And they did see some differences in those responses. But they really found some interesting CD8 T cell clones that were between the two groups that really seemed to be sort of the persistent clones that they wanted to look at further. So that's already sort of interesting to start thinking about. It's not just the Th2 cells, but we've got CD8 T cells here. And they specifically found two subsets of CD8 T cells, one that looked like a resident memory CD8 T cell, one that looked like an effector memory CD8 T cell. And these cells were found at highest levels in people with more severe disease. And they were not found so much in the healthy controls. They were found in kind of intermediate levels in people with less severe disease. So that was kind of cool. They also sort of saw that they could find these somewhat in circulation, and so perhaps they could circulate, but really they were in the tissue and perhaps repopulating the tissue after the surgery. So the real kind of interesting question for them is, okay, how are these CD8 T cells causing airway inflammation? How are they potentially causing disease? And usually when we think about CD8 T cells, at least, especially classically, when I'm teaching about them, we talk about CD8 T cells killing infected cells, using things like perforin, and some enzymes that are in granules, that we shorten to call granzymes because they're granule enzymes. We especially think of one called granzyme B, but there are others. Granzyme A is relatively famous as well. And all of these are proteases that are involved in helping to induce apoptosis in infected cells.
And the ABC, I mean, Is there a granzyme for every letter from A to K?
Yeah, I was wondering about that.
I know.
I don't know.
It
I
could
don't
be
know
sequential order, but
I think there are a lot of them.
okay.
When they started to look at these unique CD8 T cells that were in the patients with disease, they found that they uniquely expressed a high level of granzyme K transcript. So this is a granzyme that I really didn't know much about until these papers came out. Hadn't really thought too much about before
Definitely
any of
had
this.
not thought about that.
No.
Yeah.
I did find something though, Steph. It looks like there's 5 members of granzyme family in humans: A, B, H, M, and K.
Huh.
Why? I don't know.
Maybe there were other ones in like mice or something, and they some had the ortholog.
I don't know.
Fair.
Human.
And then a new one comes in.
Yeah. So, like I said, I didn't really know anything about A or B before any of this. Certainly didn't know anything about K or obviously any of the other letters because I didn't really know they existed.
Nope.
And so that was sort of interesting that granzyme K seemed to be in these CD8 T cells. They also did a bunch of studies to show that tissue granzyme K levels in patients seem to be a risk factor for having this airway disease, and actually worked as a predictive biomarker better than all of the previous predictive biomarkers like the Th2 cytokines and things like that. So that in and of itself is sort of an interesting thing to be able to use for a diagnosis. So we know that granzymes are proteases. And so they wanted to know, all right, so what is granzyme K cleaving in this case? And so they basically made granzyme K in the lab in vitro, and they took tissue lysates from the patients with the nasal polyp samples and did a precipitation, an immunoprecipitation to see which proteins could granzyme K binds, and then they did mass spec to see what granzyme K was cleaving. And they point out, we found a number of different proteins that are things that we already know granzyme K cleaves. Yay. But interestingly, the one of the things that they found was complement C3. And complement C3, of course, is a key part of the complement cascade, which is an important innate immune cascade that can lead to the different effector functions of complement. It can lead to the production of C3A, which is something that can cause inflammation. It can lead to C3B, that can help to cleave more complement, membrane attack complex, opsonization. And so the idea that these CD8 T cells were basically turning on a complement cascade by cleaving complement C3 was pretty unexpected. I often think of complement as mostly, being a very innate immune response, although, we certainly think about it being involved with antibodies. And the idea that CD8 T cells could try to turn on a complement cascade in tissue was rather new. So they also kept looking, and they saw that they could see granzyme K cleaving C2, they could see granzyme K cleaving C4, and a few other members of the complement cascade. And this is all happening in tissue, in the respiratory tract. They also kind of find that the granzyme K positive T cells make aggregates that sort of look like tertiary lymphoid organs near the vessels in the respiratory tract. And they seem to be interacting with the Th2 cells and may, in fact, these two cell types may be working together a bit. And they don't end up doing a lot more with that, although they talk about it a little bit in their discussion. And I think that that's an interest. What's going on with these tertiary lymphoid organs?
Yeah.
How are these cells working together? And things like that could be a really interesting future area to look at, whether that's through things like the C3A inflammation, whether that's, through other types of receptors on surfaces of different cell types, whether that's through chemokines. It's not really clear.
And I was wondering, related to the complement cleavage, I did read the news and views, and then I went and glanced at the parallel paper from the other group. They say that it doesn't cleave C3. So I thought that was rather interesting. So I think they both found that it cleaves C2 and C4, but one group found it cleaves C3 and the other one didn't. Which, so I just think about that and I'm like, okay, so what do we take away from that?
Yeah, I think that, so the other group was looking at different tissues. So they were specifically really thinking about the joints. And rheumatoid and osteoarthritis and synovial tissues. They do a little with dermatitis as well. And they go hardcore into the complement part of this. This one went a little more into some of the CD8 stuff, and CD8s are my thing. So
jam.
I thought that was really interesting that they both say there's cleavage of complement. They both say there's cleavage of lots of different complement fragments, but they differ on C3, which is this critical one.
Yeah, it's like the main part where all of the complements come together, and that's the effector one.
Yeah. So whether it's
I don't know,
a tissue thing or whether it's a way they set up their assays.
I was going to say maybe the
did
protein
one of them
assay
have an inhibitor that the other one didn't, or
using
lysates
like or extracts versus purified protein. Maybe there is something that is blocking that from the other group's samples and not from this one. Who knows? It's interesting though.
Yeah, absolutely. I think that it's definitely interesting, definitely a thing to figure out more going on. And so at this point, the authors switched from looking at samples from human patients and went into a mouse model of asthma, where mice get different numbers of doses of albumin along with alum and LPS. And basically what they're able to do here is they are able to use granzyme K deficient mice and show that mice that do not have granzyme K don't have the same amount of airway inflammation. And so if you get rid of granzyme K, you seem to get rid of this airway inflammation. You actually seem to, in some cases, not have eosinophils, neutrophils, all of that type 2 response being recruited into the lung. They also do, overexpression, and they show sort of both parts of this genetically. Also, really interestingly, they use a model where they have conditional knockout of granzyme K. So they're able to knock out granzyme K at different stages of the response, sort of before a first sensitization, before a first challenge, or before a second challenge. And that's interesting because you can imagine we would think about patients. We would not see them at their first sensitization. We'd see them after some challenges. So can modifying granzyme K work in those later time points? And they find that knocking out granzyme K even later on improves disease. And so that sort of highlights it as a potential place where one could intervene in some of these airway diseases. They also have a pharmacological inhibitor of granzyme K. And they use the pharmacological inhibitor in the mouse model and again show that blocking granzyme K blocks airway inflammation in this mouse model of asthma. And so it looks like these granzyme K producing CD8 positive T cells may be a key part of this tissue inflammation in the airway here. And if you sort of look more broadly with the other paper, in a few different tissue inflammatory conditions.
And I thought it was interesting also, didn't they connect it back to complement? Because in their model system, if they knocked out complement, so I think it was when they exacerbated disease by overexpressing the granzyme K. I think it was in that model. And then if they knocked out complement, they lost that exacerbation, which would suggest that if the granzyme K is mediating the disease, it's actually doing it through complement, because it's kind of connecting that full circle to their original observation. So I thought that was kind of cool.
I thought that was really cool too. And I did like that in some of the mouse models, they sort of mention again the lack of infiltrating eosinophils and neutrophils and sort of the lack of that Th2 type response. And so it sort of gives you this idea that maybe the granzyme K producing cells and the complement activation may be involved in recruiting in some of that other response or some of those other responders. Though, again, they sort of hint at that in a few places and then don't totally follow up on it. So I think that would be a cool thing to look more into later on.
So what I didn't see, though, isn't in their discussion or anything, is like, what do we know about the normal role for granzyme K, and what would pharmacologically blocking it do? Like, if it's a good target for asthma, is it going to have some side effects? Because granzyme K is important for something.
Yeah, I haven't really seen that either in any of the sort of discussion of all of this. One of the things that they do mention, and I honestly can't remember which of the three papers, if it was which paper or if News and Views talked about this, but they do show in this paper that they can compare the activity of granzyme K in cleaving complement to the activity of more classical complement activators. And it is not as efficient at activating complement. And so there's sort of this idea that was somewhere that perhaps this is sort of a inappropriate thing going on in tissue, that even a small amount of, or a relatively small amount of this complement is bad. And so maybe, because there are other complement cascades, maybe because there are other things that these T cells are doing, it wouldn't be a huge issue. But I agree with you. I don't know again, I didn't know much about granzyme K until I read this paper. So it's hard to know what blocking it would do. But I think it's a cool idea that I would be excited to read more about.
Oh, yeah.
I wonder if it would, granzymes are known to help kill infected cells. And so maybe if you have a viral infection and you're taking this anti-granzyme K therapeutic, that you wouldn't want to get sick during that time, or you'd have to be monitored or taking antivirals. So maybe that is where you could have a complication.
Yeah. It's interesting because I think that the pharmacological agent they're using blocks K, but not granzyme A or B.
Yeah, exactly.
So you'd still have a compensatory reaction, right?
I think you'd still have A and B there. That's why I'm saying I don't know what the relative role of A, B, K, M, whatever the fifth one was. I don't remember what I said, F or something. What they do, what role they play.
And I would almost wonder, how would this be in bacterial infection if you didn't have as much complement?
There are experiments done a while ago where when you infect mice with Coxsackie viruses, they get cardiac destruction, right? And it's mediated by CTLs, because if you do that in granzyme knockout mice, then you don't get the pathology, right? So, and those, as far as we can tell, those mice are okay, although they may have subtle differences. So, and I don't know what granzyme that was. Right.
Probably A or B.
Probably.
Yeah, granzyme B seems to ring a bell
in
my head. Yeah, so it has to be done, right?
Yeah.
But
now we have a reason to study K.
Is it worth doing? That's the question. Is there enough of this disease?
Yeah.
I think that there are hints between these two papers of a few different inflammatory diseases that may have a relationship here. And so particularly when you start to think about all the people with asthma, all the people with airway allergy and rhinitis, all the people with rheumatoid arthritis, we get to a lot of folks really quickly.
Yeah.
Yep.
Okay. So I wanted to just point out that this is, fundamental research that could lead to clinical application, right? And this is being done in academia. These are individuals from China. So you can't suppress this. A company will not do this basic stuff because there's no end in sight. You have to figure things out first. So this has to be done in academics, and that's what's always happened here in the U.S., and that's other countries do the same thing. So,
and other countries will get there, right? They'll get there faster if we are not.
I mean, it has to just be done. But it doesn't make any sense to kill American science because this is how it works. And the people who are killing it don't understand why it's necessary.
Yeah. Well, and this is sort of a nice example of that, because when you start a study like this, looking at T cells in nasal polyps or in patients versus healthy controls, it's not immediately obvious that you're going to see kind of a druggable target.
Correct.
Right. This is an academic problem, right?
Right. But in fact, in this case, they did find a druggable target, and you could imagine companies getting really excited based on this. But there's no way you would have known in advance that this was going to give you something that had that pharmaceutical interest.
Right.
The other interest I have is in the recirculation of these cells and what trafficking markers that they express and where they go and when they come back. So the ability of cells to migrate to different mucosal tissues is so fascinating to me. So that would be I'm sure, a whole nother paper.
Yeah, no, they're sort of hints, but certainly lots more that they can do about that. Or why did these cells differentiate to become a granzyme K producer
that would be
instead
the first
of a
question.
granzyme
What is
B producer?
The stimulant? What was the antigenic stimulation? Yeah. Probably rhinovirus. I feel like rhinovirus is just causes problems all over the place. Right?
Yeah, that's the biggest one.
But do a lot of people get rhinovirus, but not everybody gets polyps, right?
Well, right. I think probably there's going to be some genetic susceptibility because it's linked with asthma, right? And that is similarly.
It is.
Yeah.
Thank you, Brianne.
Thank you.
Okay, Steph, what do you have for us?
So we're going to stay in the airways and we're going to stick with talking about trafficking of immune cells. And so the paper I'm going to discuss today is a Nature paper published late 2024. It's called Turbinate Homing IgA secreting cells originate in the nasal lymphoid tissues in mice. The in mice I added. There's no human data in here, but really interesting immunological studies using mouse models. And I'll go through those models throughout the paper. The first author is Jingjing Liu, and the last author is Ziv Shulman. So this is out of the Weizmann Institute in Israel. And so I'll give a bit of a background, although we've been primed a little bit to talk about. Look at me.
I will tell you, I was sort of between a few different papers, and it didn't occur to me that I had sort of been, shall we say, on the nose with my paper choice until far later than it should have.
Oh, I'm like, look at us. Who are we? Alan Dove. We have puns left and right. So yes, we're in the upper respiratory tract. We know that this is a primary entry point for airborne pathogens. Rhinovirus, respiratory syncytial virus, influenza, SARS-CoV-2, you name it. This is the main portal of entry. And so you need to evolve to have mucosal lymphoid structures that initiate adaptive immune responses against pathogens, but also commensals, which we'll talk about. The gut is not the only place where commensal microbes exist and your immune response having a reaction and stimulating immunity to those commensal microbes. So we're going to talk about the NALT. This is nasal associated lymphoid tissues, not to be confused with the GALT, which is gastrointestinal associated lymphoid tissues. They develop after birth. They're non-draining, so lymph is not coming in and out. The cells are sticking there, and they are located on the base of the nasal passage, but where they're located in mice and humans is a little bit different. Humans have this thing called the Wall-Dyer's ring. I don't know who Wall-Dyer is. I'm sure he's the one who discovered this, but
could be a lady.
Could. Timing wise, it was discovered a long time ago. Statistically, probably not, but I do appreciate your inclusivity with that suggestion. Wall-Dyer's ring, located in the upper respiratory tract. So they describe it as this includes paired pharyngeal tonsils and the adenoids found at the intersection of the nasal and oral cavities. So it's like back where your nasal and oral cavities are coming together. The base of that is where the NALT is in humans. But in rodents, it's a little bit different. It's located on the soft palate on each side of the nasal passage where these paired lymphoid aggregates exist. And there's a nice picture in the paper if you're so interested in the mouse upper respiratory tract.
And sadly, Wall-Dyer is a guy.
Okay.
Oh, you looked it up?
I Googled him. He has a very long name. Heinrich Wilhelm Gottfried von Wall-Dyer Hartz.
Okay. Well, we appreciate him for his discovery, and we can think about these structures really as the places where the cells are going to meet their antigen of interest. And we want this paper really harkens back to data that we already know exists. So it was in the 80s when we were identifying IgA secreting plasma cells in human tissues, like necropsy tissues, or it's not necropsy with I do animal necropsies. It's autopsy with humans, autopsy samples. So what I think this paper is contributing is the mechanism by which these cellular interactions are happening, and using current immunological tools to prove it. You will read this paper. Maybe you all felt the same way, and you may come out of it thinking, yeah, that all makes sense. That's actually things that I maybe could have predicted based on what I know on mucosal tissues, but using these tools that I'll describe. So
I learned something. I didn't realize that necropsy was just on non-humans.
Oh, yeah. Oh, they got mad at me when I first started teaching in the vet school and I said autopsy. They're like, it's not an autopsy. It's a necropsy in animals. I'm like, okay.
Well, they don't have to get mad. They could just correct you, right?
Yes. So we are going to talk about the NALT, and one of the kind of the things that has come out of SARS-CoV-2 and this interest in mucosal vaccines is that you have a boost. You can do a prime boost vaccine strategy in the upper respiratory tract, and you will get an increase in IgA responses, suggestive that there is memory responses happening there. And so where are these IgA memory B cells coming from, and where are they being differentiated to end up in their terminal state, the plasma cell, to produce all of this IgA? And so with that little bit of introduction, I'll start to talk about the mouse models they use to be able to identify where these responses are happening. So they are going to use intact organ imaging, flow cytometry, but they've engineered a couple different mouse lines and bred them together so that we can visualize B and T cells in germinal centers, which are really the main workhorses of where these interactions are happening. Germinal centers are where B cells are undergoing somatic hypermutation, affinity maturation, class switch recombination. And in the mucosal space is gearing that towards secreting IgA. It's where you're going to find long-lived memory B cells and plasma cells that secrete high affinity. So there's this is then a CD4 T cell dependent interaction, which we'll get into. There are CD4 T cell independent interactions that produce lower affinities. So when we're thinking about vaccination inducing a high affinity response, the germinal center is what we all are focused on. How do we make them? How do we generate these robust responses that bind the antigen tightly or have great breadth to many different types of pathogens? So that's really where our story exists, is in this germinal center. So the two mouse lines I'm going to talk about, one has a Cre recombinase inserted into the AID locus. AID is activation-induced cytidine deaminase. It is the enzyme expressed by these germinal center B cells that drives antibody diversification. It introduces mutations in immunoglobulin genes during somatic hypermutation. I think this is why I love B cells so much, is Darwinian evolution is happening in you all day long. Yep. You don't have to wait till you mate and breed and make new generations of people. You have it going on inside you right now. So that enzyme is what's responsible. So you have this mouse where anytime AID is activated, the Cre recombinase is expressed. Your second mouse line carries a TD tomato fluorescent reporter that is placed downstream of a LOXP flanked stop cassette. So this setup ensures that the TD tomato is going to be expressed only when Cre recombinase is present, which is that Cre recombinase is only present if AID is activated. So when you breed those mice together, this heterozygous mice means that any B cell that's ever expressed AID is permanently marked with red fluorescence. And then you can trace these B cells over time based on this setup. They also used AID GFP knock-in mice. So the GFP is expressed from the AID locus, so the gene. So that enables this kind of real-time visualization. You're going to see GFP's green marker. So different ways of looking at B cells. This is that kind of modern immunology we're talking about to demonstrate historical concepts.
So can I summarize that? So what you're saying is the one mouse model will mark anybody who was ever in a germinal center.
Correct.
And the other one is currently in a germinal center.
Correct. Yes.
Got it.
Yes.
Okay.
So yes, you're tracing, yes, currently in a germinal center, or if the first one you said, it was in there at some time. So it's over time. Okay. So they looked at these mice without any activation, and they found robust germinal center activity, both TD tomato positive and GFP fluorescence under homeostatic conditions. This same type of germinal center activity was not observed in a comparative lymphoid structure, lymph node that they looked at. Now I have pronounced it two ways since I've started my training in science: mediastinal, mediastinal. What does the group think?
I learned the second one.
Mediastinal.
Mediastinal. Okay. I say it both ways in my head. I'm always like, oh, now I have to say it out loud. What am I going to say? So mediastinal lymph nodes are located in the mediastinal space, kind of behind the lung. So this is systemic. We're thinking of this lymph node as a systemic lymph node, not a mucosal lymph node that's interacting with many pathogens and commensals.
Is it the one that drains the nasal passage? Is that why that they're looking at that one? Or is it just a generic lymph node somewhere else? I'm not sure the placement in the mouse where that would be. Yeah, whether that might just be the draining lymph nodes. So you can look at, like, if you immunize, you can look at what's going on locally and in the draining.
Yeah, I mean, they call it the lung draining lymph node.
They call it lung draining, but when you ask about nasal draining, it does all drain there. So I'm going to say yes.
Yeah, I don't know.
But the proximity being so much longer, I think that's what they were trying to compare as something that could be actively stimulated and was local compared to distal.
Got it.
So they found a lack of this robust germinal center activity in the mediastinal. And so it starts to support this idea that NALT contains this baseline activity that the mediastinal does not. And when they looked at this in germ-free mice, they got rid of the microbes. They found a decrease in these germinal center B cells, which is neat. I think that we see the same thing in the gut. You have stimulation of, you have germinal center development from commensal microbes. So it's an interesting dichotomy. I think many people think that commensal microbes cannot stimulate affinity matured specific responses, that all antibodies are binding commensals by a polyclonal type of, maybe T cell independent. But there's work in the gut, and I think building in the nose, that no, in fact, you do have some baseline level of germinal center responses from commensals.
Yeah, I mean, it's really hard to imagine how you would have no specific responses to the commensals.
Yeah.
Yes.
Well, I mean, people thought we didn't have responses at all to commensals for a while, right? You just ignore the bugs in your gut. And now we know you have immune responses and they're critical.
Right. So, and I think the appreciation that specific responses can allow for an adaptation where an antibody binding to a virulence factor can keep commensals from getting out of control, can keep them in a niche where the immune system's happy with them, but they're not, going systemic. So, okay. So the next thing I am going to talk about is they did this, they wanted to define the spatial organization of the B and T cells in the lymphoid organs at baseline prior to vaccination, because it seems like they were very interested in this idea that you have these germinal centers prior to any of their immunization experiments. And so they did this adaptive transfer experiment where they took TD tomato labeled naive T cells and GFP labeled naive B cells and transferred them into a wild type recipient. And after 24 hours, allowed homing to these two different lymphoid tissues I mentioned, the NALT and the mediastinal. And then they used imaging to look at them and where these different cells went to. And I think they were able to observe these interfollicular regions where the T cells were dispersed, where in contrast, they're looking at the mediastinal showing the segregation of B cells and T cells. So they're making this argument that the NALT has more B cells overall and that they're more primed to interact in this T cell, B cell zone. I will say, and this is something in all of the pictures, it's not always the same mouse they're imaging. Mm-hmm. And even over time, it's not the same follicle. They're not looking at the same germinal center. There are mouse studies where you can do that. You have to, they're non-terminal, right? You have to image the same mouse, and it's laborious. So that's something I noticed. And as well, I think some of these structures could help with maybe stain some epithelial cells or stromal cells to help orient me to these structures. Because what I'm looking at here is beautiful B and T cells, and I'm just, they're inferring where these different zones are located.
Yeah. And there's always this idea when you label things and you look in an image, you see what you look for, but you don't see all the other things that are there that could be playing a role.
Right.
Right.
Right. So that was kind of the intro to the system. Here's where these B and T cells are. They're forming these germinal centers like we expect, these interfollicular regions. But now they're kind of faced with this challenge. How do you differentiate between the constitutive germinal center responses and then antigen-induced germinal center responses? Because it's going to be difficult without additional mouse models to define which are which. And so that's where they then look to a vaccine model to kind of overcome this challenge. And so they're going to look to do adaptive transfer of their reporter mice, but they're going to do intranasal vaccination before they perform the imaging. They use a specific type of mouse to get their fluorescent B cells and T cells. They use a B1/8 high heavy chain allele mouse. So this mouse expresses NP ova on its, so it is a B cell specific for NP ova. Now, NP is a synthetic chemical. It does not, correct me if I'm wrong, it does not induce an immune response without a carrier protein.
Correct.
And the carrier protein is oval albumin in this case. Mm-hmm. So you're giving the mouse a B cell that has a B cell receptor that recognizes this this antigen, which is NP ova. Mm-hmm. You're also going to give the mouse a transgenic TCR, a T cell, CD4-OT2 T cell that is specific for the ova-derived peptide presented by MHC class 1. So you have the perfect pair in this system. You have the B cell that's specific for NP ova. You have the T cell who can present the the ova.
Is OT2 class 1 or class 2?
OT2 is class 2.
I think it's class 2.
Yeah. Oh, did I say class 1? I meant
I
class
think so.
2.
But it's CD4 because you're going to provide CD4 help to the antibody, to the B cells. So it's CD4.
Mm-hmm. I didn't say CD8.
No, you didn't. You didn't say CD8.
Right. CD4 T cells, presenting MHC class 2. Okay. And so they were transferred, these cells, into the AID GFP knock-in mice. So you're tracking endogenous germinal center B cells along with your antigen-specific B and T cells. And so following the transfer, then they vaccinate with NP ova intranasally. And so the thought process is that following the immunization, the B1/8 high B cells are going to bind to NP by their receptor, internalize the NP ova complex, and present the peptides on MHC2 to allow those cognate interactions that would allow for germinal center development, because that really is a T cell dependent interaction. So what they looked at by day 5, they looked at this day 5 and day 10. They're seeing this imaging looking, they're seeing endogenous B cells were localized with pre-existing germinal centers in the NALT, but there are large clusters of these TD tomato positive B1/8 high B cells accumulated near the nasal septum. And what they suggest, they label it SED, which is the subepithelial dome. They highlight this as a key region, an early site of B cell activation. And I kind of think of the subepithelial dome maybe like a Peyer's patch, because you have these specific cells migrating there to interact to present antigen and then differentiate. And by 10, the red B cell clusters had kind of disappeared, and they're now detected within these pre-existing germinal center structures or the newly formed structures. Again, they labeled collagen here, so that maybe solved a little bit of the structure problem in regards to kind of what am I looking at here. But again, I don't believe those are the same mice on day 5 and day 10. So you're just kind of looking at...
No, they can't
sample.
be.
Right.
Yeah, it's terminal sampling.
You're kind of just taking their word for it that represents, a progression over time of what these B and T cells are, how they're interacting at that site.
Now, they also looked at the mediastinal lymph node, right? And there was a different response there, or was it similar?
Well, so with this, I'm looking at this figure, and they move on to the dendritic cell experiment. So I don't know if they showed us
I got
pictures.
a little lost because their images, they would describe what they saw, and I looked at the images. I'm like, okay.
Yeah.
Some of it I got, and some of it I had a little bit of a hard time figuring out exactly what they were showing.
Agreed. I think that wasn't always clear, and they didn't. I'm trying to look back at the paper. I'm looking at my notes. I don't think they compared it to the mediastinal for this particular experiment, which would have been helpful, right? Yeah,
because I guess I'm wondering, is it just the immune reaction just happening in the nose, or is it also more systemic?
Right. Especially as when we do influenza immunization, infection experiments, you have to put it in a solution, and the more volume we give them, the more it drains. Yeah. And we actually get a stronger immune response if we put the virus in 1 ml compared to 0.5. So I agree. I think understanding the draining responses would have been helpful.
Interesting. Okay.
Okay, so they've now set up where these B T responses are happening. They moved on to dendritic cells. I think they could have, why? I think they kind of just say, this is another cell type that would exist in the germinal center. Let's see where it is. And so they are looking at CD11C positive dendritic cells, and they've labeled them with a yellow fluorescent protein. And they are also adoptively transferring those cells in with the GFP B cells and the TD tomato red CD4 OT2 T cells. And so then they visualized them, and they could see that particularly in that subepithelial dome, you see these interactions during the initiation of the nasal immunization. And they did show that by day 10, the cells have really moved to the germinal center. So subepithelial dome, early response, then you go to the germinal center. And that early activation would be supported by the DCs and the CD4 T cells, but it is transient, as they show.
Right, because in theory, you should need the DCs to turn the CD4 T cells on so that they can go do those later events.
Right. Okay, so the next ones, they're talking about the T cell B cell boundary, and they want to see if you get if the T cells interact with B cells in this boundary prior to B cell clonal expansion in the subepithelial dome. And so again, they're doing transfer studies, same methodologies I mentioned before, GFP labeled B cells. They do go back and forth, I think, a bit between the different colors now that I'm remembering how I described the last experiment. And doing the intranasal vaccination, harvesting the nulls, and looking at different time points. And at day 3 early, they show that the B T cells are interacting in that border. But by day 5, they've progressed to the subepithelial dome and then integrate into the germinal center structures. And so I think by spatially defining where these interactions are happening, the relevance, so trying to think about like translational relevance, you're going to want to put an antigen in the area where those interactions are happening. Mm-hmm. That's what I'm trying to give them credit for as to, I think that could have been described better in the paper as to, why are we looking at these different, it is descriptive, which we need, but I think, right.
I mean, I can certainly imagine. I have a little video that I sort of show of different cells moving around a germinal center. And I feel like this supports that video very well.
Yes.
Of look at them on these different days and how you can see the green cluster move.
Yes. Mm-hmm. Yeah. I think the pictures are, I like these pictures because they're good teaching tools. I find when, giving a lecture or teaching grad students, it's nice to be able to look and see where these things are happening. So they had some other it was in their supplement, but they were trying to understand where they're coming from. So where these antigen-specific B cells that are clustered in this the dome, where are they coming from? Are they primed locally? Are they proliferating locally? And so they did a couple different experiments. They intravenously injected an anti-CD45 marker. This is a pan-leukocyte marker, so it's going to stain all immune cells. And really the purpose is that it would bind to any immune cell in the bloodstream, and you could collect blood and NALT post-injection. They did 10 minutes and see what's labeled. And it showed that all the B cells in the blood were labeled. Good. We put it there, but the B cells in the NALT were not. So it confirms that those cells reside there. They're not coming from blood. They're local. And they did some also dye injection studies. They intravenously injected some dyes to prove that point as well. So they asked about class switch recombination, which is the ability of a naive B cell to class switch to IgG, IgA, IgM. There's 5 different classes, but the main ones they're focused on is IgG and IgA. And under homeostatic conditions, the germinal center B cells in the NALT, they were primarily IgG2B with low amounts of IgA. But in the germinal center B cells in the mediastinal, they had virtually no IgA. So a little bit in the NALT. It's really following vaccination that you start to see that the majority of them in the NALT class switch to IgA, but in the mediastinal, they do not. They mainly express IgG. They don't go into it, but it is likely the cytokines that are expressed at these sites that are allowing for IgA class switching. These would include TGF-beta, IL-10, IL-5, IL-6. In the gut, retinoic acid plays a role. I'm not sure in the nose, maybe. So it's very tissue dependent. This is very nice classical immunology of IgA and IgA biology. And they're showing that to be true in the nose as well. So they switched gears a bit. They wanted to track CD4 T cells in the NALT. They say that they play a critical role in germinal center formation. So they adoptively transferred red fluorescent labeled CD4, these OT2 T cells that again express that transgenic TCR specific for ova. And then they immunized and put those into AID reporter mice. And they looked at various time points with this kind of same type of microscopy that they were looking at before. And they found early T cell activation. They harvested the NALT tissues, analyzed surface markers, and found that there's upregulation in T cell activation, downregulation in things like CD62L, which is a lymph node homing receptor. So the cells are they're not moving about, they're staying local. And they also looked at things like CXCR5, PD1 to demonstrate that these are activated CD4 T helpers that are beginning to differentiate into T follicular helper cells, which are the key T cell type in the germinal center to boost those responses. And okay, let me see where we're at. Okay, so that was T cells. They also, so they wanted to look at the responses of the cells to nasal vaccination. They wanted to know if the MHC class 2-1 B cells, where they display ovapeptide, was critical for this T cell help and differentiation of the T cells to the T follicular helper cells. So they isolated TD tomato or the B18 high B cells expressing the BCR for the NP from mice that expressed normal MHC2 or mice that had a genetic knockout of MHC class 2. Mm-hmm. And when you knocked it out after intranasal infection, and then you look at the NALT, you could find that when MHC2 was intact, you had these robust interactions. But if you did not, it it was required for T cell differentiation and T follicular helper cells. In the absence of it, there was no TFH cells detected in the NALT. So very similar to the gut and other places where we find this to be true. It is happening in the nose.
So this is sort of like what you were saying at the beginning. Yes, we knew that was going to happen, right? So it's kind of is very predictable, but nobody had ever asked it.
Nobody had
this particular surface. So we just didn't know whether in this NALT, whether it would still follow the same mechanism.
Yes. And I think one of the drivers is this idea that we want to create intranasal vaccines. Mm-hmm. We don't understand why maybe the responses don't last as long. And so diving deep into the cellular responses was key.
Yeah, because the nasal vaccines are not working as well as we'd like them to be able to. So one of the possibilities was that this didn't work the way it does in other tissues. And maybe that was an explanation. Right. But that doesn't seem to be the explanation.
Correct.
I think does seem to work the same way.
Yes, it is. I mean, it depends on what you're measuring. I think memory, like Vincent has talked about before, even though it's transient, they're memory cells that stay local. There are T cells there. So we're not used to measuring mucosal samples. So I think assessing what are we measuring is one of the things that probably we need to improve. So they go into, they asked about pre-expansion of CD4 T cells. I thought this was kind of interesting. They basically, with these experiments, I won't go through them in detail, but they suggest that you need to have a robust CD4 T cell pool to be able to stimulate B cells in the germinal centers, which didn't exist at baseline. However, they had germinal centers at baseline. And so I don't know if it's a function of their system, which is totally synthetic. It's NP-OVA. Yeah. And if they actually pulled out commensal specific CD4 T cells. So there's a little bit of a disconnect here because they're saying that at baseline there's not enough T cells to make germinal centers, but there were germinal centers there.
Yeah, I think
I don't know how you guys interpreted that.
I think that those are probably the commensals.
Yeah.
The germinal center is happening with commensals that are sort of less OT2 dependent and dependent on other T cells that they didn't put in that had been pre-expanded.
Right. And they mentioned this, the NALT of a mouse is only like 1 to 2 million cells. And so any one B cell that's specific to a commensal is going to be 1% of that. So I just wonder if it's a detection, like it would be difficult to test that outside of this like artificial NP-OVA system.
What's the goal by comparison? How many cells?
Oh, there's so many. I mean, I don't have a number for you, but the whole intestine, you get millions and millions of cells if you isolate the whole thing.
Okay.
We just isolated an intestine from a ferret and we got so many cells. So if you're looking at small sections like just the ileum, it's going to condense down.
Yeah. But the whole nasal cavity just has 1 or 2 million?
The NALT, they describe.
The NALT.
Not, we're going to talk about the turbinates in a second, but just the NALT is only 1 to 2 million. I'm going to skip through. They were looking at antigen-specific cells outside of the NALT, like where do they go? And I think generally they say the bone marrow, and that's pretty much where I would think they would go. Maybe there's 1 or 2 in the gut, but after an immune response, you're going to have some local cells that stick around, but a lot of them are going to go to the bone marrow. So we've talked a lot about the NALT, but now we're going to transition a little bit to where these cells go after they're stimulated in the germinal center. Think about the gut. Where do they go after they're stimulated in a mesenteric lymph node? They're going to the lamina propria. There's inductive sites. There's effector sites. Effector sites are where the cells are going to go. They're going to set up shop and they're pumping out tons of antibodies because they're little antibody production factories. And where these cells go is the nasal turbinates, which are different in mice and humans. Mice are much longer. Ours are a little bit, ours are shorter. But these IgA plasma cells are migrating there and they quantify the proportion that are in the nasal turbinate and the bone marrow, these OVA-specific B cells that are BLIMP1 positive. BLIMP1 is a transcription factor for plasma cells. And they find they're in the nasal turbinate. they're secreting IgA, and they are specifically going to be expressing certain trafficking markers that got them there. They wanted to understand, to prove that they are trafficking from the NALT. They did these ablation studies where they did cauterization. I'm trying to remember. It was like a high temperature cautery tip. Can you imagine like the preciseness?
Poor thing.
To like go up there and
in the mouse nose.
Yes.
They're very tiny.
Very tiny. But what they did was that they ablated the NALT, and they found that in those animals, there were no IgA plasma cells in the nasal turbinates. So if you remove where they're coming from, they don't make it to the nasal turbinate. And
can you just like help us visualize when we say NALT, it's basically, we talked about in a human, the Wohl-Dyer's ring where there's the tonsils and the adenoids, and it's kind of where you're connecting the back of your, palate to the nasal passage. Yep. Now, in a mouse, we're talking about the NALT, and they can go in and they can cauterize it. And there's this little place where they're doing the immune reaction, right?
Yes.
Now, where are the turbinates related to that? And is it analogous to say it's like our sinuses? So it's the place where there's air exchange, or am I imagining that correctly?
You are imagining it correctly. That they describe three different in the mouse. There's kind of like three different sub sections of the turbinates. And we don't have those same subsections, but it is where the air is being is that how you described it?
Yeah. So like if we breathe in now and you think about where that air goes, it goes through some passages in your nose and then back down through your trachea into your lungs and then back out. And where the pathogen's going to be, like the first place they're going to get exposure to is when they come into your, nasal cavity and where the air goes. So I guess it makes a lot of sense that that's where you would want to put those cells. If they're making those antibodies.
Yep. Yeah. Yep. So it is. It's just like what is lined all up through here. These are like
they have a picture in at the beginning, and I had to reference that a lot.
Yeah.
In order to see, oh right, this is where the NALT is. Oh right, that's the turbinate. Oh.
And it's side on. So if we cut our head in half, you have some sinuses like around your eyes and under where your cheekbones are, like. I don't know all of it, but in humans. And so mice have a similar thing where it's kind of like in the back of the head or like in the nose and below the brain and above the palate. So there are these air cavities basically, right? And then they're kept moist, I'm assuming, with lots of mucus. But that is a major location for pathogen entry, right?
Right. It is. And it makes total sense. It goes along with the other mucosal sites of the body where you're going to put those plasma cells. And they kind of end the paper talking about what markers, I mentioned that earlier, for the cells to traffic, they have to express or not express certain integrins or receptors that bind chemokines and other ligands that to help you move through the tissue. So they talk about P-selectin or PSGL. I'm trying to remember what the GL, something like P-selectin.
It's glycan or I mean, P-selectin is CD62P.
Right. And they gave an anti-PSGL1, and they found that the cells, they dropped significantly, which indicates that PSGL1 it's dependent on those cells trafficking out of the NALTs into the nasal turbinates after vaccination. And what paper wouldn't be complete without an RNA sequencing experiment, which they do. And they find different markers that are upregulated, but the one they draw attention to is CCR10. This is a chemokine receptor 10 that's expressed historically in literature. It's on all IgA plasma cells. And so once those cells come out of they go to be a plasmablast. Okay, then they move to the tissue. They become a plasma cell, fully differentiated. They're expressing CCR10, and they migrate towards CCL28. And they show in the paper that in the NALTs you will find, or in the nasal turbinates, you will find CCL28. You also find it they're showing this. Oh, let me see here. I see. They block CCL28 and they see a reduction in the transfer or the trafficking of those cells. So it follows it checks all the boxes for what we know about IgA trafficking, CCR10 expression, migrating towards CCL28 secretion from the tissue of interest. And so all of the players are there for an immune response. And I think for a nasal vaccination strategy, it is going to be about the antigen, is going to be delivery. Everyone's nose is different. Everyone has different commensals in their nose. If you have damage, if you smoke, all of those things alter the way antigen can be delivered. But what I think this paper shows to us from proving what historically had been shown with less modern immunological tools is that the NALT maintains baseline germinal center responses under homeostatic conditions. Then you stimulate with the vaccine. You have this differentiation of the germinal center. It's T cell dependent, which supports the trafficking of IgA migrating to the turbinates while they'll secrete IgA. So that is the summary of the paper.
They didn't look at memory cells, right?
I don't think so.
They did not.
No, they didn't. I mean,
because that's something you said a long time ago, that we don't know if there are actually memory cells in the NALT.
Right. I know that in different mouse studies, they've looked at memory B T cells in the lungs. I think it is less studied in the NALT.
Yeah. And so that could be part of the vaccine question as well, is you could perhaps, from everything we're seeing here, you're making kind of a primary response. But then what happens with a memory response and what happens with recall?
Because traditionally nasal infections get crappy memory, right?
I mean, I guess. Do they? I don't know. That's...
Well, mucosal in general, that's what everyone says. Mucosal virus infections, you get poor durability of memory. Yeah. And so, a vaccine has to somehow overcome that.
I think it's interesting. I actually think some of the vaccine data would support that there are robust responses. For example, LAIV is the live attenuated influenza vaccine. The reason it doesn't work well in adults is not because of a lack of ability to generate memory. It's because individuals have pre-existing antibodies that block the vaccine from replicating. That's why in kids it works great, because they don't have a bunch of IgA up there to block it.
Yeah.
Rotavirus vaccines and poliovirus vaccines, I think, work similar. Well, I know polio is a little bit different, but rotavirus, you stimulate an anti-rotavirus immunity in the gut, it protects kids from diarrhea. So I think it's a problem with pre-existing immunity at mucosal sites that makes these vaccines harder in adults, and is why they work in kids. But I don't think it's because the immune system is incapable of generating robust memory.
Well, but do we know in those cases, in the cases where it's working well, are you getting both a mucosal and a systemic response? Yeah. The mucosal response gives you sort of the location. The systemic response gives you the memory. And if you only made one, would you not get the memory?
Maybe infections limited to the nasopharyngeal cavity don't really matter. It's when they get into the lungs, then you have your memory cells and everything.
That's a good point. Maybe trying to limit infection up here is just, you're not, right, it doesn't get to the lung where it would cause you.
And it may be there's not enough cells, like you said, right?
Right. I don't know. I thought that the nasal cavity was actually important because there's, in some parts of it, there's a very thin layer between that and your brain. Right. So that's you do want to protect that site.
Yeah. I think from are you going to die of lung failure clinical side, right? But what you're describing, you do need to protect the blood-brain barrier from pathogens. And so,
well, it's just that cribiform plate that's
teeny tiny,
thin, right? With the nerves going through from the olfactory.
Yeah, because that's the other thing that I thought was really interesting is those nerves. The endings of the nerves are actually basically balled right into your airway to sense things like smell and things like that. But that leaves them quite vulnerable to infection. Yeah,
but I don't know how many pathogens actually get in that way. I think it's
surprising.
Pretty well protected.
I would try to go in that way if I were a pathogen.
Yeah, but you're not. So that's the thing. Pathogens don't think like us.
I know.
So the thing is, people used to think poliovirus went in that way for years.
Oh, really?
They thought, and they tried spraying aluminum salts in the nose of kids to prevent polio. It didn't work, of course. But
Wow.
Will going in that way basically make you much more sort of neurotropic, which is
True.
not going to be super useful in terms of transmitting later. So maybe
depends
it's a
on
bad,
how you want to transmit,
totally,
right?
you're right. Totally right, because the virus needs to spread, right? It doesn't need to get into any particular tissue. If you got just in the brain, that would be the end of it. It would never spread in the
I'm going to
population.
think it at you.
That's totally correct.
But it depends on how you decide to if you're a virus, which I'm anthropomorphizing, how you want to spread, right? Different viruses do it different ways.
But not in brain, though. Only in animals, if they eat each other, they will spread in the. But in humans, neural entry is a dead
Normally,
end.
you
Viruses
don't
don't
eat
get
each
out.
other.
No, we're not
But the
eating each other's
gut,
brain.
like Brianne said, the virus has to get in the gut. And these people never thought of that. They didn't think of
Yeah.
that. The other thing, I have this chart that I show in one of my lectures. It's got disease and persistence of antibody and all the systemic infections, 30, 75, 40, 25 years where you can detect antibody, right, in these people. And then mucosal infections, 12 months, 30 months, 3 months, 12 months. Nothing is like years.
But
So there's some reason.
Are
right?
they bleeding them? I mean, because you're
Yeah,
not going to see a
unfortunately-
mucosal response in
Yes.
blood. I think all those cells are just in the gut or wherever.
These are definitely serum
Yeah.
antibodies, yeah. Yeah.
So
I
cool.
don't know.
We learned a lot about the respiratory tract today.
So this confirms that there are the germinal centers in Nault, right?
Yes.
This is an important study because of that, correct?
Correct.
All right, good.
Now I want to know if there are CD8 T cells in there.
I'm gonna say
Yeah, they
yes.
only looked for 4, right? They only looked for CD4s.
So would you expect there to be resident CD8s?
I wouldn't necessarily expect there to be.
a resident.
Or
Right?
no, I guess they probably would be, but I'm thinking more with the other paper talking about this potentially pathogenic CD8 T cell subset.
Yeah.
That they hadn't really looked for it. They didn't know about until they just did a full panel of what was there. Here, we don't know what cells were not labeled that may be doing cool stuff.
So tell me, if you have an immunologically naive person that hasn't seen a particular pathogen, let's say a lung infecting virus, would there be CD8s there yet specific for the virus? No, right? Because it hasn't seen it. But would there be naive CD8s?
Probably would be some
Low
going through the
level?
blood.
But not resident yet.
No, not resident yet.
But then once you have an infection, then you have some resident memory, right? Okay.
But I wonder. I only know. I don't know the T side as well. But commensals exist. They're stimulating resident B cells, I don't know. I bet you there's people. You remember when Shane Crotty did that paper where they did the swabs?
Yeah, we
way
talked
back?
about that
We
here.
talked
Yeah,
about
yeah,
that. Did they look at T cells in that paper?
Yeah, I think so.
Yeah. So we can see if there were some CD8s back there.
That's episode 94, guys. Let's see, we do one a month, right?
Yeah. When's 100?
So this is, what month? This is June. So July is 95, August 96, September 97, October 98. December is going to be 100.
Yep.
What are we going to do? Gotta do something cool.
I don't know. We have
Should
to
do like a roundup.
do something different, right?
Yeah.
Yeah. But don't forget to check out the boosters too. We do those on the alternating
Yeah.
weeks.
Do you want to plug that? Because they're being posted. So tell us where you went.
Yeah. So Brianne and I went to the Society for Excite Biology meeting last year, and we recorded a whole bunch of sessions. And then I went to AAI this year, and I recorded several more. And then I also had Shane and Crowdy in my office, and we did that. So they're all going to be coming out over the next 6 months, 5 months. And then hopefully I can continue to do more. And if you guys want to, do that too.
Awesome.
Yeah.
All right, that's Immune 94. You can find the show notes at microbe.tv/immune. If you have any questions or comments, immune at microbe.tv. And if you enjoy these programs, we'd love your support. Microbe.tv/contribute. Cindy Leifer's at Cornell University. Cindy Leifer on Blue Sky. Cynthia Leifer on Threads. Thank you, Cindy.
Thank you.
Steph Longle is at Case Western Reserve University. Stephanie Longle on Blue Sky. Thank you, Steph.
Thank you. It's great.
How many years do we go back, Steph, when I first ran into you
20.
with that milk farmer that you used to podcast with?
I did. There was a dairy farmer who was very scientifically inclined and wanted to do a podcast about, the science of then agriculture, plants, animals. And so we did that, and he was a fan of you. And we started.
That was the 2000s. That was like early 2000s.
I started
right?
my PhD with Linda Safe in 2013. So it was around that time. Yep.
Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. This is my immunology course here. I learn things and then I teach the students in the virology course. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.